My name is Dr. Sofra, and I'm thrilled to be at the PAMI conference. I will be talking today about acne and skin disorders. A little bit about my background first. I have a doctorate in neurophysiology from London University and a doctorate in clinical psychology from New York. So, molecular dynamics of acne and skin disorders. Acne afflicts about 79 to 95% of adolescents. However, also individuals older than 25 years old have about 40 to 54% of facial acne. Acne persists into middle age in 12% of women and 3% of men. And it comes in whiteheads, blackheads, papules, pustules, nodules, etc. There are different types of acne types. Here you see a healthy skin. Then here you see the whitehead, the blackhead. This is what a papule looks like and a pustule looks like. Papules look like rash without visible pus. Pustules are blisters or pimples containing pus. Comedo, it's a hair follicle by keratin and skin oil kind of mixed together. Whiteheads and pimples are the closed comedos, and open comedos is the blackheads. Acne occurs at the skin sites at high density where there is sebaceous glands, such as the face, back, and chest. There is increased sebum production, keratinocyte hyperproliferation. Inflammation is a very important part of acne, and altered bacteria colonization with Propionibacterium acnes. The major pathophysiological factors are likely to be an androgen-induced increase in sebum, qualitative changes in sebum, reduced levels in linoleic acid, increased levels of lipid peroxides, increased ratio of saturated, monosaturated fatty acids. Hormones is a major aspect of this. The environment, pollution in the environment, neurologic and inflammatory mediators, and lipid metabolism. Another aspect of this is the Propionibacterium acnes cells. These are gram-positive bacillus rod-shaped bacteria that give a positive result of the gram test that prefer anaerobic growth conditions. And this is where you see the gram cell wall, and basically the gram-positive test has a peptoglycan, very large membrane here, as you can see, compared to the gram-negative test where the peptoglycan membrane is very small. So there is a whole different, basically, structure of the skin that gives the gram-positive test. There was a study that compared adult and adolescent acne. The adolescent acne is more common in males, unlike the adult acne is more common in females. And severity varies from mild comedonal to severe acne, whereas in adult acne is primarily inflammatory. This can occur in adolescent, but not as much in adult acne. And hormonal influences are actually more common, according to the study, with the adult acne. Why? As we're aging, cortisol increases and other hormones decrease. And cortisol is responsible for a number of problems on our skin, including the acne, but also inflammation and wrinkles and other conditions of the skin. Environmental pollution is a very significant contributor to acne. There is evidence from non-industrialized societies that the incidence of acne is much lower than in urban areas. So contaminated air full of dirt and debris contaminates the skin, leading to acne. Exposure to airborne pollutants, particulate matter is one of them, volatile organic compounds, ozone, nitrogen dioxide. That also is linked to pigmentations. Acne affects both Asian and Caucasian women, but Asian women have a higher prevalence of inflammatory acne. They also report that their symptoms are exacerbated during periods of high air pollution. And this is the references that are associated with chloracne is based on environmental pollutants that can result in a variant of acne called chloracne. Chloracne is caused by systemic exposure to certain pollutants in the environment and contaminated industrial waste and contaminated food products. The pathogenesis of chloracne may be related to the imbalance of epidermal stem cells. The only way to control chloracne is to prevent exposure to pollutants that induce chloracne. Now, acne and inflammation. The acne is very connected to inflammation, especially with the adult acne, and confirmed association between sebaceous lipid synthesis and inflammation. Evidence of a higher level of pro-inflammatory interleukin human sebocytes, the cells that compose the sebaceous glands. Lipid compounds, prostaglandins, also play an important role in the inflammatory response that are also linked to human sebocytes, specifically prostaglandin E2. Sebaceous glands and how they produce cytokines. Cytokines are interleukin 1A and B, tumor necrotic factor A, interleukin 6, and interleukin 8. And basically, this attracts inflammatory cells and creates this problem that is expressed in terms of acne. This is the normal skin. And this is the uninvolved acne skin, and this is the papules. And this is another study that has highlighted the inflammatory markers. This is, again, normal skin, and this is the acne skin. So you see there's a very difference, it's a big difference between the two types of skins. As you can see, Propionibacterium acnes interacts with the innate immune system and promotes inflammation. And this inflammatory process activates inflammasomes. That's a protein complex that regulates cytokine activation. So this basically comes from many different ways, many different components that interact to create inflammation and therefore the acne genesis. There is an upregulation of the caspase gene expression, induces production of MMPs, which degrade various components of extracellular matrix proteins. It stimulates antimicrobial peptides, defense peptides, which are part of the innate immune response. The innate immune response is the first type of response of the immune system, and then the adaptive immune response is more specific. It basically attacks the specific bacteria or viruses, etc. The pathogenesis of acne, then it becomes a comedone, accumulation of keratin and sebum, inflammatory Propionibacterium acnes that proliferates, and of course, then you have the scarring that follows after the acne has been expressed. This study modeled acne in vitro. They isolated the human sebaceous, the Phylosebaceous infundibulum, and they added interleukin 1A that caused basically hypercornification. And that's what happens with comedones. And this could be blocked by 1,000 ng by ml interleukin 1 receptor antagonist. So basically, when you reduce the inflammation, the acne expression subsided. Therefore, the infundibulum is capable of synthesizing interleukins, which is cytokines. Now, adding a 5 ng by ml epidermal growth factor caused a disorganization of the keratinocytes of the infundibulum and resulted in rupturing, similar to that seen in more severe pupillin grades of acne. So basically, this study found that hair follicles contain many Propionibacterium acne cells, DNA, in acne patients, as well as normal skin controls. So basically, this study is claiming that many Propionibacterium acne cells are not particularly prone to acne, except for younger teenagers. And they found that the male controls with Propionibacterium acne cells, they were highest at 15 to 19 years, whereas female controls with Propionibacterium acne cells, the highest was both in 15 to 19 and again in 40 plus. The Propionibacterium acne cells were the same in both acne patients and controls that were aged 15 to 29 years. This is a very interesting study because here it basically states that Propionibacterium acne cells are not the only reason for acne. Acne is a conglomeration of many different things that includes environmental factors and inflammation. Propionibacterium acnes has been found to be one of the main causes of sarcoidosis. Sarcoidosis disease characterized by the growth of tiny collections of inflammatory cells. We did a study, a clinical study. We had 8 subjects, 2 male and 6 females. There were dermatologists from around the world that offered their clients. And we used a new technology that came out of London University and has extremely low energies that have resultant frequencies from 0.25 to 10,000 hertz. We took pictures, before and after pictures, and we sent them to judges who evaluated the level of speed repair, the degree of repair before and after treatment. And these were the scores for the 8 subjects before and after the treatment. As we can see, there is a significant difference. The scores were from 1 to 10, with 1 the least repair, 10 the highest speed of repair, and the highest degree of repair. And again, here you see the difference. Here is the before, and these ones are the after. As you can see, there was a significant difference. The results were significant, 99.9%, very highly significant. This is the before, and this is the after, before and after, just different aspects of the same person. This is the before, and this is the after 3 treatments. Obviously, with such severe adult acne, you do need more treatments. We only performed 3 treatments. And this is before, after the first treatment, and after the 3rd treatment. So the results of this clinical trial indicated age-independent acne repair in our 8 39 to 86 years old adult patients of different ethnicities. The number of treatments required was related to the chronicity and severity of the lesion, and more chronic severe lesions required more treatments. This is another clinical case on herpes. This is the lady before, and this is after 2 treatments. As you can see, there is significant healing. As you can not see, it was much worse in real life. Here you can see it a little better. You can see the inflammation here and the damage of her skin and how it has completely repaired. It's the same lady after 2 treatments, 86-year-old with herpes zoster. This is our international research board. With this, I would like to conclude my lecture, and thank you very much for your attention. Thank you.